Hello everyone, welcome to our channel Biodiversity and Conservation. I am Nivedita Jena from Ambika Prasad Research Foundation. So today we are going to discuss about the Artemia salina. So let's start. What is Artemia salina? Artemia salina is a highly specialized crustacean, also known as sea monkeys or brine shrimp, a simple geological organism. Here is the scientific classification of the species, in which the kingdom is Animalia, phylum Arthropoda, class Brachiopoda, order Anastraca, family Artemidae, and the genus Artemia, and the species is Artemia salina. And this was first described by Carl Linnaeus in his Systema Naturae in 1758, and this was based on a report by a German scholar who had found Artemia at Leamington, England. Then here is the morphology and ecology of Artemia salina. So the brainstem body consists of three segments: the head, thorax, and abdomen. And it has three eyes, 11 pairs of legs, and its blood contains hemoglobin, which is also found in vertebrates. Um, The adult Artemia have an elongated body with two stout complex eyes, a linear digestive tract, and sensorial antennules. Distinguishably, the males differ from females by having their second antenna markedly enlarged and modified into clasping organs used in mating. Then coming to the ecology, so in nature, the life in salt lakes, they are almost never found in an open sea, most likely because of the lack of food and relative defenselessness. And so it is a species endemic to the Mediterranean, but um, found in lakes and ponds with high salinity. It can tolerate large amounts of salt, likely up to 300 grams of salt per liter of water, and can live in quite different solution of seawater such as potassium permanganate and silver nitrate. So commonly iodine, which is frequently in salt for human use, is harmful to this species. Coming to the life cycle of Artemia salina, in this slide we can see the four stage of brainstem. One is the umbrella stage and the cysts, and then naupli and the juvenile. Um, they start out as eggs, and the eggs will only hatch if environmental conditions are right. The ideal temperature must be around 30 degrees Celsius, which is similar to 86 Fahrenheit, and the water supply plentiful and soil concentration is not too high. If these conditions are not fulfilled, the fertilized eggs are deposited as cysts and remain dried and encased in a thick cell until they are ready to develop. So, the cyst may be immersed in water several times before hatching. Upon immersion in seawater, the bioconcave subsist hydrate becomes spherical and the embryo resumes its active metabolism. The cyst outer membrane cracks and the embryo emerges after a 20-hour interval, and then the embryo hangs underneath the empty cell and the development of the nucleus, which is also called larva, is completed. So, in the first larval stage, it is characterized by a distinct brownish orange color. Yeah, in this slide we can see, and a red nucleus eye in the head region, and three pairs of appendages, that is the first antenna, which is the sensorial function, and the second antenna, which is the filter feeding function, and finally the mandibles, which is food intake function. And the ventral side is covered by a large labrum, food intake for food intake, and the digestive tract of the nucleus is not in contact yet with the external medium, and after 88 to 12 hours it molds into the second larval stage, um, and after completing the 10th larval stage, its morphological and functional changes start occurring. In males, their antennae grow and develop into hooked claspers, while the female antenna degenerates into sensorial appendages. In this slide, we can see the hatching of brown string in the given clip. So somewhere a question may come to our mind that why Artemia salina is used as an idle organism for experimental assessment. So it is because of their easy hatching from dry cyst and their year-round availability, and mostly it can be used to observe, control, and manipulate a life cycle under laboratory conditions during a period of less than 2 weeks. So it is the key reason of being used brainstem or Artemia salina as an idle organism for several experimental or toxicological experiments. Here is the importance and uses of Artemia salina. So first of all, Artemia salina species individuals, especially in stage Nauplia, are used in aquaculture as live food for commercial growth of many species of fish and shellfish. And the resilience of these animals makes them ideal for testing samples in the experiments, and enhance the importance. And the Artemia salina is one of the organisms used regularly for toxicity testing of various chemicals, and together with other bodies formed by decomposition, salt lake buttermilk used to treat various rheumatic and gynecological and also endocrine diseases, etc. And the salt water and more from lake contain hormones released by Artemia salina and treat several human diseases, it is used to treat gynecological diseases, ovarian failure, infertility, etc. And it is also used in several biomedical assessments, and it is used as an antimicrobial activity of anti-cancer drugs in nanoscale analysis, etc. I hope this presentation will be informative for you all, and thank you so much for listening me.